Hello everyone. So it's 6:05. So we can start the session. I'll keep on admitting anyone who comes in late. So I'll share my screen. Can someone unmute and let me know if I am clearly audible and if the screen is visible?
Yes, you are audible and screen is also visible.
Okay, thank you so much.
Thank you. Thank you.
So we'll start with the session. So, yes. So, hi, my name is Aishi. I'm a PMRF research scholar from the Department of Chemistry, IIT Bombay. And today is the second live session of this course, Applied Environmental Microbiology. Today, we'll also be discussing some of the questions, some of this year's current assignments that you guys must have solved and already submitted, and some of last year's questions. And hopefully this solving session will help you in the future also to solve some of the other questions. And while we'll be discussing, we'll also try to talk about the newer topics that are introduced in the questions. So moving on to the first question, I'm changing my slide. One second, sorry. Yeah. So the question is, consider a situation wherein a bacterium is the only organism present in the environment that is conducive for growth. Consider the following statements and choose the best option. Sorry guys, there'll be a little bit of interruption in the middle because I'll have to admit people as and when they come in. Yeah. So consider a situation wherein a bacterium is the only organism present in an environment that is conducive for growth. So consider the following statements and choose the best option. So what do you think is the best option? Anybody? Anybody would like to unmute and answer? I am audible, right? Guys, I'm audible, right?
Yes, you are
Yeah,
audible,
okay,
but
okay.
I am in traveling. That's why I couldn't
Okay,
be
okay,
able to
sure.
walk in.
Okay. So others can also unmute and answer if you want to. Otherwise, I'll explain. So the answer will be the bacterium will split into daughter cells which will mature and replicate. So it's in the question. It's already given that the it's in an environment that is already conducive for growth. Conducive is like the environment is well made for growth, that it is perfect for growth. So that's the normal procedure that a bacterium will follow. It will split into daughter cells which will further mature and replicate. So while we're on the discussion of bacteria splitting into daughter cells, we'll talk a little bit about how bacterial cell division occurs. So as we know, bacteria is a prokaryote. Prokaryote means primitive, and pro means before, and KRI means nucleus. So as we know, prokaryotes don't have a nucleus. They have a freely floating nuclear material, and they don't have a membrane bound proper nucleus as we see in eukaryotes. So this is how a prokaryote looks. This is a prokaryotic chromosome. Prokaryotes have a plasma membrane. Have a plasma membrane, and then they have this prokaryotic chromosome, and then the prokaryotic chromosome duplicates inside the prokaryote, and after the DNA duplication has occurred, the cells divide. Wait one second, I'll accept. Someone has joined. Yeah. So after the DNA has duplicated, the cells will also divide, and the cells will basically elongate and divide. So this, and then two daughter cells will be formed. This process is known as binary fission, mitosis, and it's one of the simplest processes of cell division. So according to the question, if the bacteria gets the necessary nutrition, necessary resources, then the most easy, then they'll go into what they should do, that they'll further mature, they'll replicate, they'll split into daughter cells and follow the procedure again. So the correct answer will be the bacteria will split into daughter cells which will mature and replicate. Okay. Moving on to the next question. The two 3 phosphoglycerate molecules are converted to pyruvate with accompanying synthesis of 2 more ATP molecules, and this is known as, anybody, this is from glycolysis. So anybody would like to try this out? Anybody can unmute and try this out. This is from glycolysis. I think it's already covered in the chapters, in the lectures. Anybody? Okay. So the correct answer is pyruvate formation and ATP generation. So this is how the glycolysis process looks like. We start with hexokinase and we end up with pyruvate, and then the pyruvate moves on to the Krebs cycle. But before that, the question was that the 3 phosphoglycerate molecules are converted to pyruvate. So these are the 3 phosphoglycerate molecules in this step, in the between the 7th and the 8th steps, and these are converted to the pyruvate end product, and there's 2 ATP generation in between. And the question was pyruvate formation and ATP generation. Obviously, because pyruvate is formed and the end product at the end of this process is pyruvate, and there's 2 ATP which is transformed into 2 ATP. Also 2 water molecules also come out in the between steps. So this is also very self-explanatory. If you go and see the glycolysis process, what gets transformed to what and what are the enzymes basically catalyzing the process, you'll be able to answer these. So yes, the answer will be pyruvate formation and 2 ATP generation. Moving on to the next question. Which of the following reactions has most energy available for growth and replication? Anybody? The meeting, right? Yeah. Okay. Anybody can unmute and answer. Please do. We'd like to make it an interactive session. So anybody who's in a position to answer, please do. And it can be wrong. It's fine. We are all discussing only. Nobody?
Aerobic oxidation.
Aerobic. And why do you think aerobic? Just answer. It's whatever you think. Why do you think aerobic has the most energy available? Okay, I'll explain. So yes, you're right. It's aerobic oxidation, and that's because aerobic oxidation actually produces 18 times more energy compared to the anaerobic process. Because in the aerobic you have the glycolysis, then you move on to the Krebs cycle, then you go to the oxidative phosphorylation. So all combined it generates 38 ATP, while anaerobic only generates 2 ATP using only 1 glucose molecule. So anaerobic very definitely produces much less energy than the aerobic respiration. And with oxygen obviously, in case of aerobic we use there's oxygen in the atmosphere. So with oxygen, organisms can easily break down glucose all the way to carbon dioxide. They go through a glycolysis, then they move to the Krebs cycle, then they move to the oxidative phosphorylation, and the entire process releases 38 ATP molecules. The 36 I think are made inside the mitochondria. 2 ATP molecules are probably made outside the mitochondria. We'll have to check that, but total 38 ATP molecules are produced, which is much more than anaerobic oxidation. So that's the reason why aerobic oxidation will have produced more energy available for growth and replication. So moving on to the next question. What is the, one second, somebody is, what is the most unique factor in a eukaryotic cell that distinguishes it from a prokaryotic cell? Anybody? This easy. We discussed it in the last session also.
Option 3.
Yeah, it has a nucleus. It has a nucleus with a nuclear membrane having the genetic material. Obviously, eukaryotic cells are much more developed than prokaryotic cells. They have a properly developed nucleus with membrane or surrounding it, and that is why this is one of the most unique factor that separates the eukaryotic cell from the prokaryotic cell. So looking at the differences between prokaryotic and eukaryotic cells very quickly. So prokaryotes are referred to as simple and they generally are unicellular organisms, while eukaryotes are much more developed, complex and multicellular organisms. In the prokaryotes, they don't really have a nucleus. They have their freely floating genetic material. They don't have a proper nucleus. What they have is a nucleoid, which basically means like a nucleus, but it's not an exact nucleus. And they do not have any membrane bound organelles. Among the organelles, they only have ribosomes. The other things they don't have. The nucleus of prokaryotic cells contains DNA that is small and basically circular. We know in eukaryotes the DNA is inside the nucleus, and it's also in a chromatin form. Once the cell starts dividing, they get condensed into chromosomes, and that's when they are visible under the microscope. And prokaryotic cells have less parts compared to eukaryotic cells because they don't have all these organelles like Golgi body or mitochondria and stuff. And some examples of prokaryotic cells are bacteria and archaea. Eukaryotes are plants, animals, everything. So here also we can see the difference. Nucleus is absent, nucleus present, circular DNA, lack of membrane bound organelles. For prokaryotic cells, we have the binary fission that I discussed. They divide via binary fission or mitosis, which is a very simple procedure. But for eukaryotic cells, they divide via mitosis. The somatic cells divide via mitosis. The germline cells divide via meiosis. And so both of these are a little bit more complicated procedures than binary fission. And these are the differences. What are the similarities though? They both have cell membrane, they both have cytoplasm, they both have ribosomes, obviously, because ribosomes are required for protein synthesis. And however less developed a cell can be, it cannot survive without protein synthesis. So both have ribosomes, and obviously both have genetic material, which is the DNA. So that's the difference between prokaryotic and eukaryotic cells. Moving on. In a dendrogram, the same color lines show the bacteria with similar characteristics. Is it true or false? Anybody? Anybody would like to try?
True.
True.
Why do you think it's true? Okay. I'll have to admit, please. Okay. I think it might be false, and I'll tell you the reason why. But before that, we'll move on to see what a dendrogram basically means. So dendrogram is basically a tree diagram, as you can see here. It is used to visualize and classify taxonomic relationships. It consists of stacked branches, like these are called clades. One second, these are called clades. And at the lowest level, there will be these clades further break down into smaller branches. And at the lowest level, there will be individual elements, and then they are grouped according to attributes into clusters, with fewer and fewer clusters at the higher levels. And the end of each clade is known as a leaf. And the arrangement of the clade reveals how similar they are to each other. Two leaves in the same clade, like if there are two leaves, these two leaves in the same clade are more similar to each other than two leaves in another clade. Like this and this leaf will be less similar than these two leaves. And this basically, the height of the branch will show how close the data points and the clusters are. So I'll describe a few dendrograms before moving on. So dendrograms can be shown in a variety of ways. So this is moving on to the next slide. This is a type of dendrogram. In this dendrogram, this basically indicates the order in which the major vertebrate groups appear, with the most primitive animals first and the most evolved at the end. And this diagram doesn't have any time scale. It is simply depicting the general relationships between the groups of animals. This is one of the simplest dendrograms that you can find. Then we can move on to this dendrogram. It is a little bit more complicated. This diagram is quite similar to the previous one, but here you will notice that one branch has additional branches, like this mollusks, annelids, and arthropods. And this indicates that the mollusks, annelids, and the arthropods are more related to each other than the other groups of animals. These are the leaves from this clade. So these are more related to each other than the other groups of animals, and they all come from a secondary branch. This is yet another version of vertebrate evolution. In this one, you can see the evolution of specific traits that were major evolutionary developments marked by these red bars. Example, the first vertebrae, the development of an internal skeleton, 4 limbs, and the amniotic egg, etc. Notice that the amniotic egg separates two groups. You see, there are two groups. The one containing the crocodiles, the birds, this one, and the other with the mammals. This indicates that the crocodiles and birds are more closely related to each other than they are to the mammals, which are the primates and mammals basically. This is another version of the one that I showed just now, using some different animals groups but depicting a similar manner, a similar thing basically. And notice that this one has the base on the left instead of the bottom. So this also is very similar to the last one, just depicted in a little different manner. This is also another version of the vertebrates. This one also depicts the crocodiles, the dinosaurs, and the birds that's coming off separately from mammals. You can see the crocodiles and birds are separated from how the mammals are, and it also shows the dinosaur thought to be more closely related to the birds. So they are saying the dinosaurs are probably more closely related to the birds than to the mammals. This is yet another, a little more complicated, another way of depicting a dendrogram, and this is another dendrogram with more detail. In this one, the mammals are further divided and into the monotrims and etc., and these are basically divided into major groups, and then we have the marsupials, the placentals, etc. So you can see basically you can rotate the groups also. In this case, like in this case, if you rotate the if you light the placentals over here, then the marsupial at the bottom, it basically will mean the same thing because they will be leaves of the same clade, more similar to each other than the other branches. So basically, the dendrogram is basically used to depict the evolutionary relationships among the animals in the order how they are classified. These are some of the dendrogram images that I wanted to show. Now coming here. So in here you have a dendrogram. This is one of the slides from the lecture videos. So in here you have a dendrogram which has been represented in a tree of life format, showing that each of these trees represent each of a particular kind of microbe, basically a particular very distinct from each other. The closer to the closer they are to each other, the more clustered they will be. Like all the blue ones have been clustered together, for example, and basically the same color represents that they might be belonging to the same phylum. So like we can say with a good level of certainty that even though they are two clustered, the two bacteria clustered closer, for example, the yellow are probably proteobacteria, and even if two lines are very close to each other, we might come under the impression that overall they behave very similarly. That's what we might think, but they might be very different as well. So in this particular picture, two lines that are very close to each other and have the same color might be very different in their behavior and in their genes. They might be belonging to the same phylum, but that does not mean that they are very similar. They might be showing differing characteristics with respect to their behavioral behavior basically, and the genes that they have. So that's why when we say the same color, they are basically under the same category, but still they might be showing, if it's like if you are comparing two bacteria from two different colors, then obviously they are much different from each other, but even bacteria having the same color might be very different in their genes and their behavioral strategies. So that's why in that question I think false will be the answer, but I get why you might think it's true. The same color lines will be much similar than they are to the other microbes, but same color lines will be much similar as compared to the microbes having different color lines, but still just having the same color or just being clustered together doesn't mean that they'll have very similar characteristics. They might be from the same phylum, but still they'll be differing a little bit. They might be differing a little bit in their characteristics. Okay, so moving on to the next question. MCRA gene is linked for methane production in methanogens. Anyone?
True.
True.
Yeah, true. This is true. So basically, methanogenic archaea, they produce approximately, I think, 1 billion tons of methane annually, and their biological whatever is happening inside, it's basically mostly unknown. And this is partially due to the large phylogenetic and phenotypic diversity of this group of organisms, which inhabit, I think, various anoxic environments, including freshwater sediments, anaerobic digesters, then intestinal tracts of ruminants, all these. And also the research into this topic is mostly hampered because it's very difficult to cultivate methanogenic archaea. When you're researching on something, you need to cultivate those things in the lab, right? And you need to make environment conducive for their growth. Like we work with bacteria, we'll let the E. coli bacteria grow in the lab, but it's very difficult to cultivate such methanogenic archaea in lab. Therefore, biodiversity studies, like the studies that we have till date, mostly rely on 16 srRNA phylogenetic studies, or this MCRA, which is basically a subunit MCRA, and which I think MCRA, it encodes the alpha subunit of the methyl coenzyme M, or methyl COM reductase. And these genes are basically molecular markers for the detection and the phylogenetic analysis of methanogens. Like as for example, we know the 16S rRNA has been conserved in most categories of organisms. that is used for sequencing. You must have heard about 16S rRNA sequencing. It's used very much in phylogenetic studies. Similarly, this MCRA, which is the alpha subunit, which basically uh, encodes the alpha subunit of the methyl coenzyme M reductase, it is basically also used as a molecular marker. So this enzyme is made up of 2 alpha MCRA and beta MCRB and gamma MCRG subunits. And this entire holoenzyme catalyzes heterodisulfide formation between coenzyme M and coenzyme B from methyl coenzyme M and coenzyme B, and the subsequent release of methane. So yes, the question was, it's linked for methane production in methanogens. Definitely the answer is true, because it is one of the subunits of the enzyme that basically carries out the process. So this is a structure, it's a diagram showing how the entire methane synthesis methane production works out. So I'll be uploading all these slides, so you need not worry, you'll be able to go through these slides later as well. One second. Yes. Yes. So moving on to the next question, which of the following is not a prokaryote? This is also very easy. Anybody?
Fungi, protozoa.
Yeah, these two are not prokaryotes. Bacteria and gram-negative bacteria, these are basically bacteria, which are definitely prokaryotes. So the fungi belongs to the group of eukaryotes, which includes different types of species like yeast, mold, mushrooms, and it can further be classified into further 5 true phyla, like it's mentioned here: Zygomycota, Ascomycota, Basidiomycota, etc. And there's a recently described phylum, which is the Glomeromycota. We did not study this or the Chytridiomycota before. I think the previously only Zygomycota and Ascomycota and Basidiomycota were known, but this Chytridiomycota and Glomeromycota recently described phyla of fungi. It's a eukaryote, definitely. Protozoa are also considered to be single cell organisms, which are eukaryotes, and they possess cell nuclei, and they also share some common characteristics with animals such as locomotion and heterotrophy. And protozoa are abundantly present in environments that possess high levels of moisture and soil. So they occupy different stages of tropic levels. So obviously archaea and bacteria, these two fall under prokaryotes. Fungi, protists, and definitely plants, animals, these all come under more developed eukaryotes. Moving on to the next question. Sorry, this is another little bit between the showing the difference between fungi and protozoa. So even though fungi and protozoa both are eukaryotes, there are some differences that we can learn about them. So these are fungi are mostly multicellular eukaryotes, protozoa are unicellular eukaryotes, and fungi can move via flagella, air, water. Protozoa locomote, that is one of the characteristics that they have that are very similar to the animals, like we also use locomotion and movement, right? So protozoa locomote by flagella, cilia, and pseudopodia. And fungi cell wall generally possess chitin basically, while protozoa cell wall possess pellicle, which helps in their protection and locomotion. And these are some of the examples of fungi and protozoa. Moving on to the next question. The range of electron microscope is 100 micrometer to 1 nanometer. Anybody? True or false?
It's true.
Yeah, correct, it's true.
True.
The range of electron microscope is this. It's true. It's much better than optical microscope that we use, but then it has its own disadvantages also. If we are doing biological samples, we cannot really do it. Live biological samples are not possible under the electron microscope. It will damage the sample basically. But if you want to visualize live biological samples, we want to visualize what is happening inside by staining the various things, like we can stain the cytoskeleton, we can stain the nucleus. If you want to do that, then we definitely rely towards the light microscope. So electron microscope is also of two types: transmission and scanning. And light microscope, that's the most common microscope used to visualize biological specimens. So what are the differences? In case of light microscope, obviously light is the illuminating source, and in case of electron, there's a beam of electrons that is basically the source. And in light microscope, specimen preparation takes usually few minutes to hours. Correct. We just need to, if we are doing live cells, we basically just culture cells and we can take it and keep it on the stage and we can visualize the cells. If we are doing with dead cells or fixed cells, we can basically add paraformaldehyde to the live cells, And this once you add the paraformaldehyde, 4% paraformaldehyde, the cells get fixed. Basically, they are dead. After that point of time, they are dead. They're not live cells as such, but they get fixed in that whatever morphology they had, whatever was happening at that point of time. Even if some cells were dividing at that point of time, they'll be fixed in that morphology only. So that's how they get fixed. And then we add Triton X. Triton X basically helps in bursting of the cells, so we can visualize easily. We can, and after that we add the dyes basically to stain the insides of the cells. We add a dye to stain the nucleus, we add a dye to stain the cytoskeleton, whatever we want. So these things help us to fix, the paraformaldehyde helps us to fix the cells in their current morphology, and Triton X basically helps us to helps the dyes that we add later to get inside the cells and label the basic different parts. So specimen preparation takes a few minutes to hours. Maximum it will take you a few hours. You can actually prepare a sample in a day and visualize the same sample in that day only. But in case of electron microscope, I haven't done personally, but I have heard also that it usually takes a few days to prepare a sample. And only, as you can see over here also, only dead or dried specimen can be seen. Live cells cannot be seen under the electron microscope, because basically the beam of electrons will damage your live cells. And then in case of light microscope,
Pardon?
sorry,
Excuse me.
yes,
That live cells can be seen in the electron microscope.
but
Why?
live
Because
cells,
live cells and semi-live cells like viruses, we use it to screen that.
yeah, viruses, yeah, definitely viruses are not exactly live organisms. Viruses, yeah, even viruses can be taken from any like, if you can actually take up water from the pond and you
Cells
can
are
see
living.
whatever is inside. Sorry. Yeah, viruses are not
We
exactly
take.
lives. Viruses need a host to survive. So when we talk about viruses, viruses are not cells exactly. Viruses basically need a host to survive. When I'm talking about cells, I mean proper cells like mammalian cells or bacterial cells. Like viruses are not cells. Viruses infect cells and then they basically require a cell to survive. So, and microscope, electron microscope, when you're talking about electron microscope, you can take, for example, you can take a sample of pond water and you can visualize it under microscope. So any organisms that are present under in that water will be seen. And yes, viruses can also be seen, but viruses are, so that's a specimen that we're taking from outside. If you want to visualize what's happening inside a live cell, I don't think that's possible in a proper electron microscope. It might be possible in a cryo-electron microscope, which is another hi-fi instrument, but normal scanning or transmission electron microscope can't really visualize life
Normally,
cells.
we'll
Yes.
use very small rings.
Okay.
We'll immerse that rings into the cell trap.
Sorry,
Cell trap
small
will
what? Small wings. What did you say?
rings, small, very minute rings.
Okay.
That is made up of ghee. We used to dip it to the cell tap and keep it in the, under the electron microscope. We
Okay.
used to see many live cells and all.
Live cells. What live cells? Like which kind of live cells have you seen? Have
Live
you
cells
seen
means
mammalian?
in the plant cell itself. We
Okay.
used to see all microorganisms and all.
Are you sure those are live cells? Like, are you able to see the anything happening inside? Because what I have heard is that you
Of
cannot
course, of
properly
course.
do because
Of course,
live cells will get
after
damaged.
that they will, after that they will die. But
Huh.
even fungi also can be seen under the microscope.
Yeah.
All
What I
fungal,
meant. Hmm.
hmm.
What I meant is
Even
that
fungal,
the samples will get
fung,
damaged. That's what I
fung,
meant.
just what fungal cells and fungal micro organelles can be seen under the microscope, electron microscope.
Okay. Okay. But what I have always heard that when you're trying to, sorry, anybody
I personally did
disturbing?
that.
Huh.
That's why.
Huh, okay, but they eventually they'll get damaged. No, you cannot. They'll not like, because the electron beams. It will be very strong
They'll
for the
get
cells.
damaged.
Huh,
Damaged,
huh.
but viruses can.
Viruses, plant. Okay, if you have done with fungi and plant, I can still agree.
Right.
Viruses are not exactly cells, because viruses need cells to survive. So viruses, even I have seen people, people I have seen personally, people take pond water and then they have tried to see what are the specimens, what is there in that pond water. So yes, they have got very good images of viruses also. But viruses, I don't consider viruses as cells. But if you have done fungi and plant, then I guess it's possible. But then at some point of time, the samples will be damaged. What doesn't have, it doesn't happen like that in light
Fungi we
microscope. Yeah.
did. Fungi we did.
Okay, okay. Then okay.
Fungi
Then
also we can.
okay. then it might be possible you can try to visualize under the electron microscope, but at some point of time it will get damaged. But this thing doesn't happen under light microscope. Obviously, if you're using a very high powerful laser, your samples, if you're using a particular dye, your sample might get bleached. In case of optical, like in case of microscope, if you're even if you're talking about confocal microscope, which uses a pinhole to focus on a particular plane, if you're using a very high power laser, and if you are basically uh, uh, the power of the laser is more than 50% or so, some parts might get bleached. But then we have done live cell imaging under light microscope, and cells don't get damaged as such, because light is not that harmful. But in case of electron microscope, even if you try to do live cells, then the beam of electrons will damage the okay, so you have done, that's great. So
Yes, that's right. That's right. What you said is correct. After exposing to beam of light, they will die.
Okay, 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 huh. Yes, so that's what I meant. So yes, so it's not that, sorry for that, it's not that it's not possible. You can try to do it, but after exposing it to the beam of electrons, it will die, because that's very strong and harmful for the live cells. And for light microscope, that's not the case. Viruses I have seen, I have not seen plant or fungi, but it's great that we can at least try to capture something before the cells die. But yes, so that's that. Viruses need cells to survive. So viruses, I don't think those can be called under live specimen. But anyway, but definitely the electron microscope resolving power is much better than light microscope. Magnification is much better, and basically the range is much better in an electron microscope than a light microscope. But if for my work, we basically see what is happening inside the cell, and we need the cells live. Like I have done FRAP, which is basically fluorescence recovery after photo bleaching. So what we do is that we label a particular thing in a cell, and then we bleach that particular portion, and then we try to see how much of the fluorescence is recovered. Like what is the basically what is the dynamics, or what is the fluidity in that particular area. So for that we need live cells. We cannot do with dead cells, because dead cells will not move, dead cells will not show any that kind of recovery. So for that, light microscope is a winner, definitely. But in case of magnification and resolution and everything, obviously electron microscopes, everything is much better than light microscope. Yes. So moving on to the next question. Gram negative bacteria has an additional layer of periplasmic proteins than the gram positive bacteria. Anybody?
True.
Okay, true. So this thing is, I have found actually very contradictory points regarding this. Somewhere people say that gram positive bacteria don't have any periplasmic layer at all, but in some areas I have seen that gram positive bacteria does have a periplasmic layer, but it's very it's not that much as compared to gram negative bacteria. Gram negative bacteria have a more prominent layer of periplasmic proteins, and in some other cases I have seen that some gram positive bacteria might have periplasmic area, some gram positive bacteria might not. But gram negative bacteria definitely has a much more prominent layer of periplasmic proteins as compared to gram positive bacteria. So when we talk about the differences among the two bacteria, gram positive and gram negative, uh, gram positive bacteria has a plasma membrane. It might have a very less prominent periplasmic space, and then a very huge wide peptidoglycan layer. In case of gram negative bacteria, in case of gram negative bacteria, gram negative bacteria, there's a plasma membrane, there's much more prominent layer of periplasmic space, there is a thinner peptidoglycan layer as compared to gram positive, and there is an outer membrane of lipopolysaccharide and protein. So these are the basic differences of a gram positive and gram negative bacteria. And in some cases they say that the periplasmic space is absolutely absent in gram positive, but some gram positive bacteria might have a periplasmic space as well. So the gram positive cell wall has a thick peptidoglycan layer, as I showed before, while gram negative bacteria has a very thin peptidoglycan layer. In case of the gram positive, gram negative, it comes from a very particular process of staining these bacteria, which is called gram staining. I'll talk about this gram staining in detail, but before that we'll know that gram positive bacteria, after the process of gram staining is done, gram positive bacteria is shown to be stained in purple color, while gram negative bacteria is shown to be stained in pink color, and I'll tell you why. In case of the teichoic acids, it's absent in gram negative bacteria, but gram positive bacteria has teichoic acids. And gram positive bacteria does not have any outer membrane, while gram negative bacteria has an outer membrane. And the gram positive bacteria basically stains in purple color because it retains the crystal violet stain. That's the first type of gram staining. It retains the crystal violet stain, while the gram negative bacteria does not retain the crystal violet stain. That is why it is not purple. We add another stain after that, and the gram negative bacteria takes up that stain. We'll come to that later. Gram positive bacteria are less resistant to antibiotics. Gram positive bacteria are more resistant to antibiotics because they have an outer membrane, so that's why they are more resistant. And the layers of peptidoglycan, it has a single peptidoglycan layer, gram negative, but gram positive has a multi-layered peptidoglycan layer. And lipopolysaccharide concentration, as I said, more in gram negative, less in gram positive, so thickness, resistant to drying, etc. So I'll move on to gram staining a little bit. So this is the entire protocol of gram staining. So what happens, you add a primary stain which is crystal violet. You add it to the slide. There's a bacterial smear on your slide, and you add this drop of primary stain or crystal violet to your slide. So both the types of bacteria will take up the color of the stain, that is purple. Then you add iodine. So the iodine is basically works as a mordant. It makes the dye less soluble, so it adheres to the cell walls. So basically when you're adding iodine, you're basically ensuring that the dye attaches to the cell wall more properly. So still now, even now we have both the bacteria purple in color. Then we add a decolorizer, which is alcohol. So what happens when you add alcohol? The alcohol decolorizes and washes away the stain from the gram negative bacteria. So now the gram negative bacteria is colorless, and the gram positive bacteria has retained the stain, which is purple. Now safranin, which is another counter stain, that is the second stain that we are using in this protocol, and basically a counter stain. It is added to the slide, and it is pink in color. So now the gram positive bacteria, they had retained the crystal violet color only, and the gram negative bacteria now takes up the pink color of the safranin. So this is what happens. So after the entire procedure is done, you can see under the microscope, and the pink colored bacteria that you can see will be gram negative, and the purple colored bacteria will be gram positive. So this gram staining and differentiation process is based on the differences in the cell wall structure and the composition of the bacteria basically. So bacteria having cell walls with a thick layer of peptidoglycan will resist decolorization of primary stain and appear violet or purple. So gram positive bacteria have a multi-layered peptidoglycan wall, so they'll resist the decolorization that comes from alcohol, and they'll appear purple. Bacteria having a thin peptidoglycan layer like the gram negative bacteria, they'll basically they'll have lesser cross linkage. They'll lose the primary stain as soon as you add the alcohol, and then gain the counter stain, that is a safranin, the pink color. So this is what happens. This is basically what happens in the gram staining procedure. Moving on, mitochondria has its own genetic material, and that is distinct from the genetic material inside the nucleus of a eukaryotic cell. Is it true or false?
False.
You think it's false? Anybody?
True.
False, ma'am.
Okay.
True.
So it is actually true. Mitochondria does have its own genetic material, and basically people have hypothesized that mitochondria at some point of time during the process of evolution, mitochondria might have been an aerobic bacteria because they have their own genetic materials. They might have been living independently at some point of time, but with the evolution process, the eukaryotic cell has basically engulfed this aerobic bacteria, and now this aerobic bacteria that has come inside the eukaryotic cell has developed to become a mitochondrion. So this hypothesis has come from the fact only that mitochondria has its own unique genetic material that is distinct from the nucleus of the eukaryotic cell. And right now, I don't think mitochondria will be able to live independently because it has some of the genome of the mitochondria has actually moved to the nucleus as well. So right now, I don't think mitochondria will have any capacity to live independently. But at some point, this is a hypothesis that mitochondria used to be an aerobic bacteria, and it has a DNA of its own. So yes, mitochondrial DNA versus nuclear DNA. Inside the matrix of the mitochondria, there is mitochondrial DNA. Nuclear DNA, we all know, is inside the nucleus. It stays in chromatin form, that is coiled form. During cell division only, it forms the chromosomes, the condensed structures of chromosomes. So if we talk about the differences between the mitochondrial DNA and the nuclear DNA, the mitochondrial DNA is the mitochondrial genome. Nuclear DNA will be the cell's genome, the entire cell's genome. The mitochondrial DNA is double-stranded but circular, but the nuclear DNA, as we know, it's double-stranded linear. And mitochondrial DNA will be like freely floating in the mitochondrial matrix, but in nuclear DNA, it's found in the nuclear matrix, fixed to the nuclear envelope During cell division, it forms condensed structures called chromosomes. Nuclear DNA is obviously surrounded by the nuclear envelope. It's tightly packed with histone proteins. Histone proteins are basically basic proteins which help in the formation of chromosomes, but mitochondrial DNA, they are not associated with any kind of histone proteins. Then these are some factual data like nuclear DNA might be containing 20,000, 25,000 genes. So, and including 3 mitochondrial genes. While mitochondrial genome, they have 37 genes encoding 13 proteins, 22 transfer RNAs, and 2 ribosomal RNAs. Some of the mitochondrial genetic material has shifted to the nuclear genome. That's why in the nuclear DNA has 25,000, almost 25,000 genes, which is very believable because it's the nucleus of the cell, and it also has 3 mitochondrial genes. Then we all know nuclear DNA will have coding regions and non-coding regions. There will be exons, introns. Mitochondria is not differentiated as such. They don't have non-coding DNA regions. And mitochondrial DNA replication is very independent from the nuclear DNA replication. Nuclear DNA replication happens during the interphase. The synthesis phase, during the interphase, the DNA duplicates, and then the mitosis starts. Mitosis, which is the cell division, starts happening. And the mitochondrial DNA is very fascinating fact. Mitochondrial DNA is maternally inherited. So whatever mitochondrial genetic material that you have, it's from your mother. It's maternally inherited. However, the nuclear DNA that you have, it's equal from both the parents. Inherited equally from your mother and father. So these are some of the differences of mitochondrial DNA. And as I said before, scientists were quite amazed to find that this organelle which is inside the cell, why does it have a genetic material of its own? Probably because it used to live independently at some point of time, but then it's now a part of the eukaryotic cell. So moving on, start codon, next question. Start codon binds to the starting sequence of DNA in the protein synthesis. Anybody?
True.
Okay, what is a start codon? Do you have any idea? Just say what you know. So start codon, so we know that we have 4 nucleotides, right? Adenine, ATGC basically. Adenine, thymine, cytosine, and guanine. So we have 4 ATGC nucleotides, and when we are talking, and these ATGC, basically these 4 nucleotides make up your entire genetic material, DNA. And when protein, these encode proteins, right? So when proteins synthesis happens, the smallest unit of protein is amino acid. So amino acids will come together and form a polypeptide chain, and then protein will form. That's another story. But amino acids, each amino acid is encoded by a 3-letter codon. So this answer of this question is actually false, but I'll explain what start codon is. So we have these 4 AGCU. It's U basically because if you're talking in terms of RNA, it will be uracil because RNA doesn't have thymine. If you're talking in terms of DNA, it's thymine. It's easier to talk in terms of uracil because as we know, according to the central dogma, the DNA gets converted to mRNA or the messenger RNA, and from the messenger RNA the proteins translation, like proteins are synthesized from the messenger RNA. So it's better to talk in U terms, uracil. So these are the 4 nucleotides, and there are the 3 nucleotides will come together to basically denote a particular amino acid. So this genetic code can be read in multiple ways depending on where the reading starts. For example, if the base sequence is GGG, AA, CCC, the reading would start from the first letter G, and there will be 3 codons, GGG, AA, CCC. So each 3-letter bunch is called a codon. That's what a codon is. So if it starts from G, then the first 3 G will be a codon, then the middle, the 3 A's will be a codon, and then the 3 C's will be a codon. But if the reading starts at G in the second position, in this example, the string will have 2 codons. Like the code, if the reading starts from the second G, then it will be GGA, then AAC, and then moving on. That's how it will move. So if reading starts from the third base like this, third G, then it will be 2 codons like GAA. According to how the reading starts at exactly which letter, that will denote the amino acids that come and attach to that basically come to form the protein. So there are basically 3 ways of reading the code of every standard genetic material. Then again, if we move to A, it will AAA, CCC, it will again be the same reading frame as the first one, GGGAA. So from this base sequence, we can figure out that there are basically 3 ways of reading the code of every strand of genetic material. Now, these different ways of reading a nucleotide sequence is known as the reading frame, and each reading frame will basically produce a different sequence of amino acids and hence proteins. When you shift the reading frame by even one letter, the codon gets changed and there will be a different amino acid encoded by that codon basically. So we have 3 reading frames, but in a double-stranded DNA, there will be 6 possible reading frames. Okay, so now what is the start codon? So you can see that there is a start shown in green over here, and it shows methionine. So when we are basically the mRNA has these nucleotides. So when the reading starts, the mRNA, there is the ribosome that forms the protein. The mRNA comes at a particular site in the ribosome, and then the reading starts. And once there will be a particular start codon, AUG is the start codon. So once the ribosome encounters that particular start codon in the mRNA, that's when it will start synthesizing the protein. That's when it will start bringing the amino acids to form the protein. There's another RNA called the tRNA. The tRNA, transfer RNA basically brings the amino acids. So the mRNA is attached to a particular site in the ribosome. It's reading the sequence. Once it encounters the start codon, that's when it will start bringing the amino acid. That's when the tRNA will start bring the first amino acid, which is the start amino acid, is methionine. Methionine is an amino acid. The start codon is AUG. You can see here A, U, and then G. So AUG is the methionine code, and the moment it figures out that AUG is here, it's the start codon, the tRNA will bring methionine, and then accordingly the sequence will be read, and more amino acids will be brought, and the polypeptide chain will be formed. So start codon binds to the starting sequence of DNA. This is a wrong statement, because start codon is present, it will be in the mRNA and it will be read by the ribosome. DNA generally has nothing to do with this, because DNA has already done its job. DNA was a genetic material, it has already converted to mRNA, and now it's the mRNA's job to, so from DNA to mRNA it's called transcription, and from mRNA to protein it's called translation. So now it's the mRNA's job. So this statement is not right. So when the ribosome is reading through mRNA, it will encounter the start codon, and then it will move on to read the the following sequences, the following codons, and there will be more amino acids coming and joining. So as we have start codon, we also have 3 stop codons. So the stop codons are UGA, this is stop codon, then UAG, and then UAA. So these are the 3 stop codons. So when you are reading in this sequence, 3 codon you have read, methionine has come, that's the start codon. And the next 3 nucleotides you have read, another amino acid has come. Next 3 nucleotides, so the next codon is read, another nucleotide has come. Once the ribosome encounters any one of these stop codons, UGA or UAG or UAA, it will terminate the process of mRNA translation to protein. So that's how it works out. So this is what happens. This is the ribosome. This is small subunit, large subunit. There's a particular site. There are 3 sites in the ribosome, EPA sites. There's a particular site. This is the mRNA. This structure that you can see, it's the mRNA or the messenger RNA. And each of these colored things, these are basically nucleotides. So each of the 3 bunch of nucleotides will denote a codon. So the ribosome is reading through the sequence mRNA, and as and when it reads through each of the codon, the tRNA brings that particular amino acid and helps it to attach to the chain. So this is the amino acid chain that has formed. It will read another 3 nucleotide, another codon. The tRNA will bring that particular amino acid and it will get attached to the amino acid chain that's formed. So the codon AUG is the start codon, as it's the first codon in the transcribed mRNA that undergoes translation. And AUG is the most common start codon, and it codes for the amino acid methionine in eukaryotes and formyl methionine in prokaryotes. But during this, the most common start codon means generally in eukaryotes it's AUG only, methionine, but some other organisms might be having some other codons, start codons. It is not exactly fixed. In eukaryotes it's generally methionine, and in prokaryotes it's formyl methionine, but there might be some variability as well. If you do more research on it, you'll find that there are some other codons also in some other variety of organisms that are thought to be start codons. And during the protein synthesis, the tRNA is the transfer RNA, it recognizes the start codon AUG with the help of some initiation factors, and then the translation of mRNA starts. So this is the process is called translation. It is not this simple as it looks in this diagram. It is a very rough diagram. It is a very complicated procedure. There are a lot of initiation factors. There are a lot of proteins coming in, going out at the same time. So if you're fascinated about it, please, you can study about the process from the Watson, the book. It's a molecular biology book. It's a Watson. It's by the author Watson. So you can go through it. It's very complicated. The process is not as easy as it is shown here. Moving on to the next question. Dispersion is an example of the passive transport in the bacterial cell. Anybody? True or false?
False.
Yeah, correct. Dispersion is no way close to passive transport in the bacterial cell. So what does dispersion mean in biology? Dispersion is basically the spreading of a population or organism away from its parents. And it's very self-explanatory. What happens, what does it mean to disperse? Disperse it basically you're spreading away from a particular position. So when the population is spreading away and when it is moving away from its parents, and they're looking, because the organisms are looking for some additional nutrition, additional resources, or any change in the environment, that's when dispersion happens. And it can be influenced by a variety of factors such as temperature, terrain, limitation of resources, anything. Animals disperse by moving because we are privileged to be able to move, and we can show locomotion and movement. Plants have seed dispersal. It gets seeds get carried away by wind, or seed dispersal can also happen by various other organisms that come to the plants. But animals, they disperse on their own via moving. And there are basically three types of population distribution. One is clumping. In here, the organisms are clustered together in groups, and there is social interaction, there is mating, and their resources are basically clumped. This is the most common pattern of population dispersion. This may reflect a patchy distribution of resources in the environment. This is where everyone stays together. Even if you're dispersing in group. Then there is random. In random, the organisms have a very unpredictable distribution. It is very rare, can be hard to determine whether truly random or largely clumped. One second. So this kind of random distribution is random dispersion is quite rare, and it has a very unpredictable distribution. In this case, species, individuals, they do not interact that much strongly as is seen in clumped. You can see in clumped in dispersion, they are actually moving in groups, so there is more interaction. In case of uniform, the organisms are basically uniformly spread out in an area. This is very typical in case of a species where the individuals are competing for resources. So if there are limited resources such as water, in case of a desert, if there is very limited water, so in that case there is a uniform distribution. Not everyone will, they will not show a clumped dispersion, because they will not be able to survive. There will be huge competition for water. So there is uniform distribution. So these are the three most popular patterns of population dispersion. Moving on. Transposable elements can jump from one location to another location in the genome. One location in the genome to another. Anybody? True or false?
True.
Yeah, this is true. Transposable elements are also called jumping genes because they have the capacity to move from one location in the genome to another. So these are basically DNA sequences, and these were first identified by this geneticist called Barbara McClintock of Cold Spring Harbor Laboratory in New York. So mostly these are of two types, the DNA transposon and the retrotransposon. What happens in case of DNA transposon? In case of DNA transposon, the one part of the DNA is excised. It is cleaved out. It's called the DNA intermediate, and the target DNA where it is looking to jump into, that comes into view, and then proper integration happens. In case of retrotransposon, what happens, that there is a DNA, one part of the DNA gets converted to the RNA intermediate, and then reverse transcription happens, and RNA intermediate and the RNA gets converted to DNA again, and then this DNA gets integrated into the target DNA. So that is why it's called retrotransposon, because the reverse transcription is happening in the middle. So these are the these are the popular categories of transposon or jumping genes, and these basically occupy half of the human genome, and it also forms a very huge fraction of the plant genomes, and these are basically ubiquitous in the biosphere, and they are also major forces in the evolution and rearrangement of genomes. So you can see, so if there's a particular gene, and if a jumping gene goes and integrates in between that gene, the gene's function will be hampered. So sometimes transposons can also be very harmful because it can lead to mutations. because if there's a proper if the proper functional gene in the genome and a transposon is going and getting integrated in the middle, so that in that way it can lead to very what can I say, mutated protein, mutated products. The protein product might not be functional. So there are various types of effects that might be caused due to the transposons or jumping genes. Okay, moving on to the next question. All enzymes are 3D structures of linked amino acids. Anyone?
True.
Yeah, true. Actually, it should it's true, I agree, but enzymes in this case they are assuming that these enzymes are proteins. Some enzymes can also be RNA. In that case, this is not a correct statement. If you are assuming that the enzyme is a protein, one second. If you're assuming that your enzyme is a protein, then true. Enzymes are, because proteins are 3D structures of linked amino acids, so enzymes are also 3D structures of linked amino acids. In that way, it's true. But some enzymes can also be RNA. So in that case, RNA are not made of amino acids. So in that case, those enzymes are not 3D structures of linked amino acids. So please keep this in mind. But if you're assuming that it's protein, then this statement is true. And that's because proteins have different levels of structure: primary structure, secondary structure, tertiary and quaternary structure. So the primary structure is basically the alphabets of the protein, like the amino acid residues. It's a primary straight line structure. What are the amino acid residues? They are coming and they are how translation occurs. The amino acids come and they properly form a polypeptide. So that's the basically primary structure. Then the secondary structure forms. So two popular secondary structures are alpha helix and beta sheet basically. So you can see it over here. Alpha helix is a very spiral type of structure and beta sheet is a sheet type of structure. So the secondary protein structure, this happens because of some hydrogen bonding amongst the sequences, and this causes the amino acids to fold into a repetitive pattern. And then this is the secondary structure. And then comes the tertiary structure, which is basically the 3D structure. It's a three-dimensional folding of the pattern of a protein due to side chain interactions. So some of the side chains are also interacting. So when the side chains are interacting, the three-dimensional thing is happening, and that's how the tertiary structure is getting seen. And then there's a quaternary structure. That's when different subunits of proteins come. Different subunits of proteins of the particular protein. There are different subunits. Like I talked about the methyl reductase protein right now, MCRA. So they have different subunits. They come together and form a protein. So when different subunits are coming together, that's how a quaternary structure is formed. So yes, these are there are more than one amino acid chain. One amino acid chain forms a subunit. So there are more than one amino acid chain. They are coming together to form the quaternary structure. They're assembling together, and these assembled subunits are called proper proteins. Moving on, any given protein will have a primary, secondary, tertiary and quaternary structure that will give it distinct properties. It's very similar to the last question. Anybody? Anyone? It's very similar to the last question. So this is also true. As I said, the primary structure is the amino acid chain or the polypeptide chain. Secondary structure will be the alpha helix and the beta pleated sheets, and the tertiary structure is how they three-dimensionally fold on top of each other due to side chain interactions. And finally, the quaternary structure, where different subunits and different polypeptide chains will come together to form a proper protein. Yes, property. So next question, decay term in the growth model is important because it, anybody? I'm still in the meeting, right? Yeah. Decay term is in the growth model is important because it, anybody? Which is the answer? You can see the answer. Anybody would still like to try? Okay, because it incorporates the removal of the microorganisms due to natural death. So before that, we'll learn a little bit about the bacterial growth model. So this is the bacterial growth curve, and the different stages of this curve indicate different stages in the bacterial growth basically. So that when it starts with a very plateau like, it's not a plateau, it starts with a very standard phase, like there's a straight line, there is no upwards movement or downward, that's a lag phase. So this is a very distinct phase. In the initial phase of the lag phase, the bacteria are metabolically active, but they are still not dividing. So in this phase, when you basically bacteria are stored in glycerol stocks in -80. So when you take out a bacterial vial from the -80 and you put it in the -80, they are preserved, but they are not dividing when they are kept in -80. So when you take, and they are metabolically also not active, but when you take them out and you put them in liquid media and then you keep it in the shaker at 37 degrees Celsius or whatever the optimum temperature for your bacteria is, mostly it's 37 degrees Celsius if you're talking about E. coli. So when you put it in liquid media and keep it in 37 degrees Celsius, at first you will not see immediate growth happening, because at that time there's a lag phase is happening, because at that point it will take some time for the bacteria to get accustomed to this new atmosphere. Till then it was in -18 glycerol, but now in 50% glycerol stock, but now it has suddenly been put into a proper liquid media with proper nutrition and everything. So at that point it will get metabolically active, but it will still not divide. So that's the initial phase, which is also called the lag phase. Then the next phase will come, which is the exponential phase or the log phase. So that is when there will be a huge rise in the number of bacteria. It's the log phase, that is the bacteria it's exponentially dividing. So for example, if you're talking in terms of E. coli takes 20 minutes to divide. So once it has crossed the lag phase, every 20 minutes the bacteria is dividing. So that's the exponential phase. Most of the bacterial division will happen over here. Then comes the stationary phase, or which is this plateau region. In the stationary phase, this growth reaches a plateau because the number of dying cells equals the number of dividing cells. At some point of time, the bacteria will come to realize that the nutrition that we had in this medium is limiting right now. So we have divided a lot, but now the nutrition that was given to us, it's limiting, and at that point of time there will be a plateau-like region. The bacteria, the number of dividing and the number of dying bacteria will be the same, and then there is a decay or the death phase, and that is shown by an exponential decrease in the number of living cells. So if you don't change the media, then if you don't add proper nutrition, then the bacteria will start dying. It's the same like if we are starving for a lot of time, we'll face issues. So that's how, if you keep the bacteria starving for a lot of time, they'll start dying. That's the decay or the death phase. So obviously the decay and the death phase in the growth curve is important because it incorporates the removal of the microorganisms to natural death. Natural death is happening because the resources have become limiting now. They are not getting proper nutrition to survive. So that's why they are being removed from they are basically dying. That's how it works. Now next question. In logistic growth model, the nutrient limitation plays an important role in determining the final population. Anyone? True or false? There are two growth models, exponential and logistic, for bacterial growth curves. So anybody would like to take? Okay. Yes, you are right. The answer is true. So we'll talk a little bit about exponential and logistic growth models and how these are different. So the general equation for the population growth rate, that is the change in the number of individuals in a population over time, will be dNdt Rn. So in this equation, the dNdt is the growth rate of the population in a given instant. N is the population size, T is the time obviously, and R is the per capita rate of increase. That is how quickly the population grows per individual already in that population. So if there are no movement of individuals into or out of the population, so we are talking about bacteria in a test tube. So obviously there are no individuals moving out or inside the population in this case. So R is just a function of birth and death rates. So if we are talking about a huge population of elephants in a forest, then the moving in and out of that population matters. But we are talking about bacteria, so there is nothing of that sort happening. So R is just a function. R is only dependent on the birth and the death rates. So when the per capita rate of increase R takes the same positive value regardless of the population size, then we get exponential growth. When R is always positive, and when there is no limiting resources, when you have infinite resources, when the bacteria gets proper nutrition all the time, there is nothing to stop the bacteria from dividing, and that's when we get the exponential. One second. That's when we get the exponential growth curve. But this theoretically seems very
That exponential growth is only theoretical, right?
Yeah, that's that
Maybe
I was
for
coming to. Yeah, that's what I was coming to. So this is possible very theoretically, because at some point of time the resources will start to will start to basically get depleted. Resources are not unlimited in the environment. So that is what I was coming to. Exponential is a very theoretical model. It happens in theory, but the logistic growth is more realistic, because obviously the nutrients will get limiting at some point of time. So if you're talking about bacteria, you have put the bacteria in a liquid media, in 5 ml liquid media, but as the bacteria is dividing very fast, 20 minutes, it's the generation time. It's doubling every 20 minutes. So at some point of time, the nutrient in the bacteria will stop being enough for the number of bacteria that has come. So that is when the logistic growth model is more realistic. So here the per capita rate of increase decreases as the population increases towards the maximum limit. So this is the logistic growth model, and this is denoted by this equation. So at this point of time there is no exponential growth as such, and then we mostly come to see the plateau region, the stationary phase that comes in the logistic growth model. So in this exponential growth, as I said, the key concept is that the number of organisms increases, population, whatever be the population size, R is always a positive. So it's a maximum rate. So that is, for instance, bacteria can reproduce much faster than humans and would have a higher maximum per capita rate of increase. So this is how the exponential growth curve will look like. dNdt, R is R max N. So in this case, R is always positive and constant. Nothing is changing. So it will lead to exponential growth. But in logistic growth, exponential growth is not a very sustainable state of affairs. As I said before, it's very theoretical, since it depends on infinite amounts of resources, which tend to, in the real world, we do not get infinite amount of resources. It's very limiting. So that's what he said also, that exponential growth is not very realistic. It may happen for a little while when there is a lot of nutrition in the media, but if there are a few individuals and there are a lot more resources, there will be exponential growth for a time being. But when the number of individuals, they divide and they get large enough, the resources will start to get used up, and it will slow the growth rate. And at that point of time, it will give a S-shaped curve. The population size at which it levels off, that will be, this will represent the maximum population size. The plateau region, that will represent the maximum population size a particular environment can support, and that will be, there's a new term, that is the carrying capacity, or K. So this logistic growth model equation is R max, K minus, somebody is joining. Okay. So R max, this is the logistic growth model equation. So it's a little different from the, there's a new term, K minus N by K, because now the carrying capacity is also taken into account. So this is the logistic model. So at any given point in time during a population's growth, this expression K minus L will tell us how many more individuals can be added to the population before it hits this carrying capacity. This is the carrying capacity. The plateau area is the carrying capacity. And then K minus N by K is the fraction of the carrying capacity that has still not been used up, that is still remaining to be used up. So the more the carrying capacity that has been used up, the more this term will reduce the growth rate. So when the population is very tiny, that is N, when the population is very tiny, the N is very small compared to K, the K minus N by K term becomes approximately K by K, or 1. So when N is very tiny, if N is almost negligible, then K by K becomes 1, and it almost gives us an exponential equation. So it fits when the population is very tiny and there are huge resources. At that point of time, it may become an exponential curve. But when K will, as and when the organisms will divide and they'll start showing more population size, then logistic model is more realistic. So that is that. The question was, the logistic nutrient limitation plays an important role. Definitely nutrient limitation plays a very important role in the logistic model. It doesn't matter in the exponential model. So this is what factors determine the carrying capacity. Basically any kind of resource important to a species survival can act as a limit. So for plants, water, sunlight, nutrients, space. For animals, important resources include food, water, shelter. All these are very important resources that organisms need to survive, and limited quantities of these resources result in competition between members of the same population. Intraspecific is within your same species competition. It will lead to competition, and interspecific competition for resources may not affect populations that are well below their carrying capacity. So when the population is very tiny, this kind of competition won't matter. So there is a huge there are huge availability of resources and your population is very tiny. So this kind of competition won't matter because there is enough for everyone. But that is when resources are plentiful. But when the population size increases, that's when the competition will intensify, basically. Moving on to the next question. Low turbidity signifies exponential phase in the logistic growth model. Anyone? So it's false. Anybody? So yes, low turbidity does not signify the exponential growth in a phase in a logistic growth model. So when we are checking the bacterial growth, we are turbidity or OD or optical density basically is one of the things that we check when we are checking if the bacteria is growing properly or not. So the exponential phase means that the bacteria is dividing very rampantly, very quickly. So low turbidity does not signify the exponential phase definitely in any growth model. Next question, alpha helices and beta strands form the tertiary structure in proteins. True or false? Anybody? We discussed
False.
false, yeah. Because alpha helices and beta strands are the secondary structure. These are the popular secondary structure of the protein, as I discussed. So this is the alpha helix, this is a beta pleated sheet. These are the secondary structure, and this happens because in the alpha helix a hydrogen bond is formed between the NH and the CO of group of the amino acid, and every winding turn of the alpha helix has basically 3.6 amino acid residues, while in case of beta sheet it basically consists of various beta strands, and these are also linked by hydrogen bonds between adjacent strands, and 3 to 10 amino acids are combined to create a beta strand polypeptide. And beta strands we generally see in fibrils and protein aggregates in amyloidosis, and these are secondary structure. This is not the tertiary structure, definitely not. So a little bit of difference between the alpha helix and beta sheet. Alpha helix, as you can see, it's a spiral structure, so it's in the right-handed coiled rod-like structure, while beta sheets they exist in an almost entirely extended conformation. There is intramolecular hydrogen bonding that forms that basically there's hydrogen bonding here, as you can see in this sketch. So that's intramolecular within the molecule. So there's intermolecular hydrogen bonding happening in the polypeptide chain, and that's what creates the and gives stability to the spiral structure, while beta sheets are formed by linking two or more beta strand intermolecular hydrogen bonds. So different molecules form hydrogen bonds, so this is intermolecular. And as I said, one turn will correspond to 3.6 amino acid residues, while here in case of this 3 to 10 amino acids are combined to form a beta strand. And beta sheet cannot be in a single chain polypeptide, definitely not, as you can see from here. There must be two or more beta strands, while alpha helix can be present in a single chain polypeptide. And some of the examples are keratin, myoglobin, and for beta sheet there are skin fibers or fibroin. So next question, for any genetic element there are dash open reading frames. This also I discussed when I was talking about start codon. Anybody? How many reading frames are there? So there are six, correct. So there are three reading frames as we discussed from any strand, but in case of genetic element, since it's written genetic element, we are assuming it's double stranded DNA, so that's why it's 6, but it should be more clear. A double stranded DNA will have 6 open reading frames, correct. So this is the previous slide that I had shown. For every sequence there will be three reading frames, and in case of double stranded DNA there will be 6 reading frames. So I'm not explaining this again because we have already covered this. Most energy rich compounds in cell primarily, anybody? Okay, so it has been seen that most energy rich compounds in cell primarily are phosphorylated compounds with ester and hydrate bonds, and one of the very like the first option is correct, and a very popular example is ATP or adenine triphosphate, adenosine triphosphate, sorry. And this has 3 phosphate groups over here, and it's a very high energy compound. So whenever we talk about energy in organisms, we talk about adenosine triphosphate, ATP. Moving on to the next question, which of the following statements is not true? Exponential stationary lag death at different phases of microbial growth on a petri dish. Net ATP generated and complete utilization of glucose during aerobic respiration is 24. The correct sequence of bacterial population growth is lag exponential stationary death, and A catalyzes degradation of the beta 1 for glycosidic bonds in the outer cellulose units of cellulose. Which one do you think is not correct? Anybody? Which statement do you think is false over here? can say. So the second question is for second statement is false over here. The net ATP generated in complete utilization of glucose during aerobic respiration is 24. That's false because we already discussed it's 38 in total. Because in aerobic respiration we have O2. The entire process shows glycolysis, Krebs cycle, and then oxidative phosphorylation. And the citric acid cycle itself produces 36 ATP molecules. And in total there are glycolysis produces 2 and the rest of the Krebs cycle produces 36. So as I said before also 2 ATP molecules are formed outside the mitochondria and the rest are formed because of glycolysis. The other 36 are found inside the mitochondria, and so it basically the entire process gives rise to 38 molecules of ATP. So the 24 number is wrong, so that is the false statement over here. And as we talked about the bacterial growth curve a few slides ago, a bacterial growth curve does have a lag phase, an exponential growth phase, then a stationary phase, and a death phase. So yes, so these are very prominent phases of a bacterial growth curve, and total cells in population can be seen, like there are, as you can see from the diagram, the sketch over here. So these are very prominent phases of the cell growth curve of bacteria. So this statement is also correct, and the sequence of statement is also correct. First it's lag, then exponential, then stationary, then death. Next question is, which kind of reaction has the most energy available for growth and replication? I think this also we covered. It will be aerobic because aerobic oxidation will have the most energy, definitely, because as I said it gives rise to 38 ATP molecules in total from glycolysis, Krebs cycle, etc. So it will be the most energetic process, energy giving process. Next is select the most suitable option. Anybody would like to try? Anybody? I'm still in the mood, right? Yes. Okay. Anybody join in? Okay. Anybody? So the answer will be 1F, that is photosynthesis will be paired with complex 3. 2 will be D, which is O2. The terminal electron acceptor is oxygen in complex 4. 3 will be B, which is the E from the torque is generated during proton motive force, and that is what happens when it's limited from E from outer to inner membrane. Inhibition site is alpha beta catalyst interface, and ATP synthase will be conserved in all forms, so that's why P5 is A. And PMF, that is the proton motive force, basically it's the electric potential between the inner to the outer cell membrane. So the final stage of aerobic respiration is the electron transport chain, which is located on the inner mitochondrial membrane, and this process have very distinct steps. So there are proton pumps that create an electrochemical gradient between the two, the outer compartment and the inner compartment. That's called the proton motive force, or the PMF. And the ATP synthase that is there, it's basically in the structure of a rotor. It uses a subsequent diffusion of protons to synthesize ATP, and the oxygen accepts electrons and protons to form water. So the electron transport chain, it's located in the inner mitochondrial membrane. So if we think about the structure of the mitochondria, the inner mitochondrial membrane is folded into structures called cristae, and these cristae structures are basically there to increase the surface area available for this transport chain. And this entire ETC, or the electron transport chain, releases the energy stored within the reduced hydrogen carriers, and basically the main motive is to synthesize ATP. So that is what happens in this oxidative phosphorylation procedure. So in step 1, what happens? A proton motive force is generated. So the hydrogen carriers, that is NADH and FADH2, these are oxidized and they release high energy electrons, as you can see from here, and protons. Electrons are transferred into the electron transport chain, and this consists of several transmembrane carrier proteins. They are different categorized into different complexes. So there are various transmembrane carrier proteins, and the electron is moving to this transport chain. So as electrons pass through this chain, they subsequently lose energy, which is used by this chain to pump protons from the matrix. So as you can see, the electron is moving through the transmembrane proteins, and with every movement it is losing its energy, and this energy is utilized to pump these protons from the low concentration to high. So basically what happens generally, any solute or any substance can move from a high concentration to a low concentration, but when you, that is along the concentration gradient, but when you want movement of something against the concentration gradient, that's when you need to apply energy. That's the difference between active transport and passive transport, right? In passive transport, substances move from high concentration to low concentration, so along the concentration gradient. But if you want to transport a substance from low concentration to high concentration, you will need an external another source of energy. So the electron is giving that source of energy. The electron is moving through the transport chain, and the protons are being pumped outside, so from the matrix to outside. Outside is high proton concentration, and still it's getting pumped outside. That's why energy is required. The accumulation of these protons within the intermembrane space creates an electrochemical gradient or a proton motive force. So that is what is PMF. PMF is the proton motive force. So this is step 1. PMF or the proton motive force is generated. In step 2, the proton motive force, this proton motive force that happens, it will cause all the H plus ions to move down their electrochemical gradient. The electrochemical gradient is obviously this way because that's high concentration, this is low concentration. So this will allow all the PMF or the proton motive force will allow all the proton molecules to protons to come down the electrochemical gradient and diffuse back into the matrix. And this diffusion of proteins is called chemiosmosis and is facilitated by this transmembrane enzyme called the ATP synthesis. So this green sketched and green sketched thing here is called the ATP synthesis. So the PMF is allowing these protons that were thrown out of the matrix to come back inside in a process called chemiosmosis, and it is using this ATP synthase to do it. So as the protons are moving inside through this ATP synthase, they trigger the molecular rotation of this enzyme. So the molecular rotation of this is basically a rotor kind of thing. So this enzyme is molecularly rotated and ATP is formed. ATP synthesis is happening. That is how the electron transport chain is helping in ATP synthesis. In the step 3, oxygen acts as a final electron acceptor. So in step 3, what happens? In order for the electron transport chain to continue functioning, the de-energized electrons must be removed. So the electrons are moved through the electron transport chain, losing energy at every step. So in order to keep this to make this function further, the de-energized electrons that were here must be removed. And so oxygen is here. Oxygen acts as a final electron acceptor. It removes the de-energized electrons to prevent the chain from becoming blocked. Because if you don't remove the electron, this chain will become blocked and no other electron will come in to throw proton out. No other proton will come in via the ATP synthesis and no more ATP will be produced. So what happens? The de-energized electrons are taken by the final terminal electron acceptor, which is O2. And O2 also binds with some free protons. So now in the matrix we have a lot of free protons. So oxygen also binds with some of the protons, free protons in the matrix. It takes electron and now it forms basically it maintains the hydrogen gradient as well. Maintains the hydrogen gradient as well. So in the absence of oxygen, if oxygen is not present, the hydrogen carriers cannot transfer energized electrons to the chain and ATP production is halted. So that's why aerobic oxidation, aerobic respiration, it produces so much of energy in the presence of oxygen. Oxygen has a huge role to play, because if oxygen was not here, there will be no terminal electron accepted to accept the electron, and this entire process will become blocked. To make this entire process will become blocked, and what will happen? ATP will not be synthesized anymore. So that's why aerobic respiration is aerobic oxidation creates the most amount of energy, because aerobic mein oxygen is present and oxygen takes the electron, oxygen takes the extra free protons, and it allows the chain to continue functioning. So these are the steps of the electron transport chain. So that's how the hydrogen carriers donate high energy electrons. Hydrogen carriers are NADH and FADH2. They donate high energy electrons to the electron transport chain. Electron transport chain located on the cristae. What are cristae? The infoldings of the mitochondrial inner membrane. And then as the electrons move through the chain, they lose energy, which is transferred to the electron carriers within the chain, and these carriers then used to pump the hydrogen ions from inside the matrix to outside against the gradient, inside the matrix to the intermembrane space, that is outside against the gradient. And this accumulation of H plus ions in the intermembrane space then creates an electrochemical gradient, proton motive force, which then allows the H plus ions to come back inside through the ATP synthase, and this basically leads to ATP synthesis. And then oxygen comes at the last step as a terminal electron acceptor. It takes de-energized electrons, it takes away some free protons from the matrix, and then it allows the electron transport chain to keep functioning and to keep synthesizing ATP. So this is the exact thing. This is also a really complicated procedure. We have tried to explain in a very simple manner. So the last complex, the complex 4, is called the cytochrome C oxidase. and its role is in the oxidation of cytochrome C. And this highly complicated molecule consists of multiple heme groups, cofactors, and subunits. And basically, this complex 4 allows the transfer of electrons from the cytochrome C molecule to the oxygen atom, that is the terminal acceptor, and then reduction of oxygen, 2H plus is taken, water is formed, and simultaneously complex pumps 4 plus H plus ions into the intermembrane space. So that is how the electron transport chain works. So what is proton motive force? I've already talked. It's a force that that promotes movement of protons across the membrane downhill in the electrochemical gradient. It is proton motive force leads to the generation of ATP, and basically that's what happens. So according to the questions, the question was matching the sequences, so these will be the correct answers. PMF is proton motive force, and ATP synthesis is obviously conserved in most life forms because ATP is required in all the life forms. So next question, which of the following is not true? Energy is consumed in steps where carbon skeleton is broken down in citric acid cycle. Acetyl CoA is a 2C molecule, 2 carbon molecule, and triggers citric acid cycle. Oxaloacetate is a 2C molecule and is essential to continue citric acid cycle, and pyruvate is necessary to triggers citric acid cycle. Anybody? Citric acid cycle is basically the Krebs cycle, the TCA cycle, Krebs cycle, citric acid, all are synonyms. Anybody would like to try? Anybody? I'm still in the meeting, right? Okay. Anybody can unmute and try. Okay. So the wrong answer is oxaloacetate is a 2C molecule. Oxaloacetate is not a 2 carbon based molecule. It is essential to continue citric acid cycle, definitely, but it is not a 2 carbon molecule. The other statements are all correct. Energy is consumed in steps where carbon skeleton is broken down, and there's acetyl CoA is a 2C molecule and it triggers citric acid cycle. Pyruvate is necessary because pyruvate is the end product of glycolysis, and from glycolysis we move ahead to citric acid cycle, so pyruvate is also necessary. So this is the citric acid cycle, and as you can see from here, oxaloacetate is a 4C 4 carbon based molecule. It's not a 2 carbon based molecule. Acetyl CoA is a 2 carbon based molecule, but oxaloacetate is a 4 carbon based molecule, and this is the entire citric acid or the Krebs cycle that comes after glycolysis. So yes, the second option is not true. Third option is not true. Oxaloacetate has 2C based molecules. That part is not true. Next is choose the most suitable traits. Yes. Anybody would like to answer? This was covered in the lecture videos by Gargi ma'am. Anybody would like to answer? You can open your lecture slides and also try to answer. Anybody? Okay, I'll answer then. The correct answer will be option 4. 1D, 2B, 3C and 4A. 1D is Delovibrio, Deloblast. 2B is Myxobacteria can be matched with slime trail. Then we have 3C, L pneumophila, that can be matched with need amoebic host to replicate, and then 4 is quorum sensing, which is basically social predator. So Myxobacteria is also kind of social predator. So Myxobacteria can be matched to social predator, but then Myxobacteria as a slime trail also, and slime trail must be matched with Myxobacteria because quorum sensing can't be matched to slime trail. Quorum sensing is also a type of social predator thing. So that's how this is the most relevant pair. The fourth option is the most relevant, and I'll tell why. So let's talk about Delovibrio. Delovibrio is basically a gram-negative obligate aerobic bacteria, and this is its life cycle basically, and it basically preys on gram-negative bacteria and not gram-positive, and they have two lifestyles: a host-dependent, highly mobile phase, the attack phase, in which case it basically latches onto the host. It goes to the periplasmic space of the gram-negative bacteria, and then it forms a structure called Deloblast. So that is why we have matched Delovibrio to Deloblast. So Deloblast, they attack the host, which is a gram-negative bacteria. Very important. They don't attack gram-positive bacteria. And the reason might be because gram-positive bacteria don't have a prominent periplasmic space. gram-negative bacteria have a very prominent periplasmic space. So what happens, the Delovibrio, it attacks the host or the gram-negative bacteria. It goes into the periplasmic space. It forms a deloblast kind of structure. It forms a structure which is called deloblast in the host bacteria, and which is a very slow growing and irregular shaped and host dependent form. And the most studied form of Delovibrio is a Delovibrio bacteriovorus, which is written here, and it's almost exclusively found in host dependent growth in nature. And in this free swimming attack form, after searching for prey, it basically burrows through the host outer membrane, cell wall, and enters the periplasmic space. That's what I said. It goes into the periplasmic space, then it forms a structure called deloblast, and inside the deloblast, the singular large flagellum of the predator Delovibrio is lost. You can see the deloblast kind of structure. It's a very vague, rough sketch. And then the host cell is killed, rapidly killed, allowing the passage of molecules from the interior of the host cytoplasm through the periplasm villi and the periplasm dwelling of Delovibrio. So it's basically like a parasite. It's coming, it's latching onto the host cell, it's going into the periplasmic space, creating a structure for its own survival, and then basically harming the host cell and killing off the host cell. And using some molecules, the Delovibrio creates a protective environment by reinforcing the peptidoglycan cell wall of the host. And now, after around 4 hours, depending on the temperature, the Delovibrio has now increased in size, and because they have taken the entire nourishment from the host, they have killed the host cell, but they have taken all the nutrition from the host, and then it divides, it replicates, and then leaves. It basically lyses the host cell and leaves with the host cell's cell wall, and it lyses the host cell wall and membrane, and then it leaves. And this newly emerging Delovibrio has a very powerful flagella to swim away. It can swim very powerfully. Then it goes and finds the next suitable host. Basically, it's a bacteria infecting another bacteria. So and it forms the structure deloblast. So that's why we have matched Delovibrio deloblast over here. So Delovibrio are also called cytoplasmic predators. Delo means leech. It's highly motile because it has now got a very powerful flagellum after coming out of the host bacteria. It is highly motile and curved bacteria. It's an obligate aerobe, and it attaches to the prey cell. It penetrates the cell wall of the prey, and it replicates in the periplasmic space. Can attack a variety of G negative bacteria but not G positive. So I think logically it's because G positive or gram positive bacteria don't have a very prominent periplasmic space. That's why it's not able to attack gram positive bacteria, but gram negative bacteria have a very prominent periplasmic space, so it can attack a variety of gram negative bacteria. So predation is not an obligatory lifestyle, but it still is a cytoplasmic predator, and it's widespread in most aquatic habitats. Next is social predator myxobacteria. As I said, myxobacteria can also be called a social predator. It also has a very complicated lifestyle, and under starvation, what happens, they aggregate into huge and elaborate fruiting bodies. And within these fruiting bodies, the cells differentiate into myxopores. So these slides are taken from the lecture slides only. So myxobacteria is also called the social predator. As when I was discussing, I said myxobacteria can also be matched to social predator, but it has also a slime trail. So what happens? This is this is Delovibrio life cycle. So I gave it last. So I already explained this, so I'll not go further to explain it anymore. I'll move on to myxobacteria. Myxobacteria leaves a slime trail when it's infecting. So that's why myxobacteria can be matched with slime trail. Moving on, we can talk about quorum sensing. So what is quorum sensing? Quorum sensing is basically the regulation of gene expression in response to the fluctuations in cell population density. And it is mostly seen in case of bacteria, where bacteria release chemical signal molecules called AI or autoinducers, and this basically this concentration of autoinducer, it increases as a function of cell density. It basically depends on the cell density. As and when the cell density fluctuates, the autoinducer concentration might also be fluctuating. And this entire process of quorum sensing can be divided into 4 steps. There is production of small biochemical signal molecules by the bacterial cell, as you can see over here, and these signal molecules are then released either actively or passively into the surrounding environment, either passively like along the concentration gradient, or actively like with usage of energy against the concentration gradient into the surrounding environment. And these signal molecules are then recognized by specific receptors once they exceed a threshold concentration, and that leads to gene regulation. So once this particular AI means autoinducer concentration reaches a particular limit or threshold, some of the receptors of these cells then recognizes these, and that basically starts a signaling process leading to change in gene regulation. And one common consequence of quorum sensing induction of gene expression is increased synthesis of proteins involved in signal molecule production. This increased synthesis is basically creating a positive feedback loop. So the molecules are getting produced, autoinducers are getting produced, they are increasing in concentration, they are attaching to the receptors. These receptors is once they attach to the receptors, particular receptor, a cell signaling process is started, and this leads to gene expression regulation. And then in the gene expression regulation, the gene corresponding to uh, the involved in signal molecule production, these proteins are more and more synthesized. Basically it's a positive feedback loop, it's sending a positive feedback, because now they are saying that we are synthesizing more proteins that are involved in signal molecule production, and this thing is going back, and then basically the quorum sensing process is more enhanced. It is forming a positive feedback loop, and that is why these quorum signals are also called autoinducers. What does auto means? Auto means automatic, what you can do, or what happens automatically basically. So these inducers are auto-inducing themselves. These are getting produced, they are going back in a positive feedback loop. That's why they are called auto-inducers. So myxobacteria I was talking about, it's also called slime bacteria because it leaves a slime trail, and that's how it's able to move via gliding. They typically travel in swarms and have molecular and containing many cells kept together in molecular signals. And the individuals of the myxobacteria strain, they benefit from aggregation and allows accumulation of extracellular enzymes that are used to digest food. So myxobacteria can be matched with slime trail. Next was the Legionella pneumophila. So Legionella pneumophila or L pneumophila is a facultative intracellular parasite, and it basically needs an amoebic host to survive. This is the life cycle of Legionella pneumophila, and these are basically shown to be proliferate in the walls of pipes in biofilms. So this is the life cycle of Legionella pneumophila. It can infect a phagocyte. It needs an amoebal host to further infect. So in case of this match and pair, Delovibrio will be matched with Deloplast, Myxobacteria will be matched with slime trail, Legionella pneumophila will be matched with C or need amoebic host to replicate, and quorum sensing is also a type of social predation technique because basically you are living in a social atmosphere. There are many bacteria coming together and they are basically acting like one single organism. They're creating more and more autoinducers, they are attaching to receptors, and the entire thing is getting carried on. So quorum sensing is also a type of social predation. So quorum sensing can be matched with social predator. So moving on to the next question, which is true for predatory bacteria? Delovibrio are predatory only under stressed environments, and social predators often behave as multicellular organisms. Anybody would like to try? We talked about both of these right now, so you can try. Anybody? Okay, so both 1 and 2 are true. Delovibrio are not obligatory predators. They are predatory only under stressed environments, and social predators often behave as, just now I said, with the context of quorum sensing. Social predator, when they are working as social predators, they work as basically one huge single organism. So that's how they often behave as organisms. So both 1 and 2 are true. Multicellular organism. So which of the following is not true for diazotrope? Diazotrope is a nitrogen fixing bacteria. So anybody would like to try? Out of these following 4 options, which one do you think is not true for diazotrope? Anybody? So the correct answer will be they are always found in symbiosis with other organisms. This statement is not true because there are a lot of nitrogen fixing bacteria which are free living, and there are symbiotic like in the rhizobium and stuff, but there are also free living nitrogen fixing bacteria. The other statements are true. So diesotropes are bacteria and archaea that fix gaseous nitrogen in the atmosphere into a more suitable, usable form, such as ammonia. And it's able to basically grow without external source of fixed nitrogen. It can basically take nitrogen from the atmosphere and fix it. And there are free living diesotropes also there, but they need a mechanism to protect dinitrogenase from oxygen. So oxygen can inhibit the enzyme which is dinitrogenase. So free living diesotropes actually need a very prominent mechanism to protect this dinitrogen from oxygen, and so that's why they are obligative. That's why they're obligately anaerobic is one solution. What is obligate anaerobe? Obligate anaerobe means they cannot survive in presence of oxygen. So in order to save dinitrogenase, free living diesotropes are mostly obligate anaerobic. They can't survive in presence of oxygen. And so obligate anaerobes are organisms that cannot survive in presence of oxygen if they are not fixing nitrogen. They live in habitats low in oxygen, such as soils. Example, Clostridium is an example. Sulfate reducing bacteria are important in ocean sediments, etc. So the last statement was false, because there are various free living diesotropes as well. So there can be two types of diesotropes: free living or symbiotic. Symbiotic is when you are in a symbiotic association with another organism. The most popular examples are rhizobia in the root legumes, and then frankia. And the free living nitrogen fixers are different. So the contrasting habitats of free living and symbiotic nitrogen fixation is explained over here. FLNF is nothing free living nitrogen fixer. So basically they are carried out by a diverse array of nitrogen fixers living in a community, while symbiotic nitrogen fixation can only be performed by a few bacteria, very popular but very rare bacteria like rhizobia and frankia, and they live in population. And the free living nitrogen fixation is supported by dissolved organic carbon in the soil. So they take, because they are in not any kind of a symbiotic relationship with any other organism, they have to get all the nutrients and everything required themselves only. So they get the dissolved organic carbon they take from the soil, and while in case of free living nitrogen fixer, but in case of symbiotic nitrogen fixer, they receive a constant supply of simple carbon compounds. So we know what is the meaning of symbiosis, right? And symbiosis means a relationship where both the parties are benefiting from each other. So the plants are benefiting from the bacteria is also benefiting from the plant. So what is the benefit that the plant is getting? The bacteria is fixing nitrogen for the plant. The bacteria is taking nitrogen from the atmosphere and fixing it for the plant. That's a huge benefit. In return, the plant is giving all these substances to the bacteria. So in case of symbiotic diesotropes, what happens? They get a constant supply of simple carbon compounds, like it's, for example, succinate. It's shown here, for example, succinate from the directly from the host plant. Again, next point is the oxygen concentration in the rhizosphere is highly variable and driven by soil structure and texture and respiration. So conversely, symbiotic nitrogen fixers are supplied oxygen at low concentrations by the host plants. Everything they get from the host plants. Nutrients that are necessary to support the free living nitrogen fixers, they are free living, so they are not in association with any kind of plant. So the free living nitrogen fixers, the nutrients they need to acquire by themselves, because they do not have a friend to give them a constant supply of these stuff. And these nutrients are delivered to the symbiotic nitrogen fixers by the host plant. Mostly the free living bacteria will get most of the nutrients in the soil or the atmosphere, but for the symbiotic bacteria it's very easy. The host plant is supplying everything. And in the diesotropes in the rhizosphere can access nitrogen from soil, and the free living nitrogen fixer, in case of symbiotically fixed nitrogen, it's delivered to by the plant. So this is the difference in the contrast. It's basically a contrasting habitat and the way these two different types of diesotropes fix nitrogen. So biological nitrogen fixation contributes around half of the annual nitrogen inputs in the biosphere, and it's an important source of nitrogen in natural ecosystems and also in agricultural systems. And one of the options was regarding the NIFH gene. So the NIFH gene, it basically encodes for an enzyme called dinitrogenase reductase. It's a component of the nitrogenase enzyme, and it also serves as a genetic marker. So this NIF gene is a biomarker, and it is used to study the ecology and the evolution of the nitrogen fixing bacteria. So So that was one of the question over here. So the NIF is genetic marker. This point is also true. They're obligatory anaerobes. This point is also true. Free living diazotrophs have to be obligatory anaerobes because they have to protect the nitrogenous enzyme from oxygen, because otherwise it will get inhibited. So all these points are true except the last point. They're always found in symbiosis. Not true. There are also free living nitrogen fixers. Next question. Match the following. There are 4 in this category and 4 in the other category. What option do you think is correct? Anyone? You can try it. It's fine. You just unmute and try. We are all discussing over here, so it doesn't mean that you have to be right. Anyone?
Option A.
Option A. 1C, 2D. Okay, 1C, 2D, 3A. Okay. You're correct. But why? I'll explain. External metal reduction is geobacter. So wait. So geobacter is an iron reducing bacteria, and this is another slide that I've taken from the lecture videos. So basically these are some of the key genera that can be categorized under the dissimilative iron reducing bacteria. They are common in anoxic freshwater and marine sediments, and they couple metal reduction with H2 oxidation. They are phylogenetically very diverse, and many other organisms enzymatically reduce metals in their environment but are unable to use this for their cellular growth. So yes, geobacter can be 1C, yes. So external metal reduction can be geobacter. Then next we are talking about hydrogen metabolizing bacteria. So hydrogen, one of the key points of the hydrogen metabolizing bacteria is that Ni is it's an essential element in hydrogen. Nickel is a very essential element in hydrogenases and must be present in the culture. So this also matches with this option. Ni, nickel is required for hydrogen oxidizing bacteria. So this is they have great metabolic diversity, the hydrogen metabolizing bacteria, and they are also referred to as hydrogen bacteria. They grow best under low oxygen, that is 5 to 10%, because hydrogenases are typically sensitive to the presence of oxygen. Next is acetic acid bacteria. So gluconobacter is a very popular acetic acid bacteria, and because in the industrial preparation of acetic acid, gluconobacter is also used. It includes the key genera Acidobacter and gluconobacter, and it includes the obligate aerobes, and it's used in the industry production, and this use the acetyl CoA pathway to produce acetate. So that's gluconobacter is then used in the industrial production of acetic acid. Then the last one that was left was D, which is to use ferrous oxidation for cell growth. So this match and pair, what she said is correct. Moving on to the next question. Select the most suitable option. NIFH gene, 16S rRNA gene, nitrogenase, and then there are 3 further options. Anybody? Anybody? At least try.
Option A.
Option A. Option A is 1A. Okay, 2B. Correct. 1A, 2B, 3C. Right. NIFH gene. This gene is very popularly transferred by horizontal gene transfer. So what is basically horizontal gene transfer? Horizontal gene transfer is the movement of genetic information between different organisms, and by this process various antibiotic resistant genes are also transferred from one bacteria to another. So these three are some of the horizontal gene transfer techniques. There's transformation when we put a DNA inside the bacteria. There's conjugation happens between 2 kinds of bacteria, F plus. There's a bridge kind of structure that is formed during conjugation, and the bacteria can actually share its genetic material via this, or transduction, when a bacteriophage, a virus which can infect a bacteria, which is a bacteria infecting virus, is called bacteriophage. A bacteriophage infects bacteria, and then it incorporates its own genetic element into the bacteria, and that is called transduction. So these are the three different horizontal gene transfer techniques, and NIFH gene is a very popular biomarker, genetic marker, and it's also transferred via horizontal gene transfer. So it can be 1A. 16S rRNA gene, as I said, it's conserved amongst various organisms, and that's why 16S rRNA sequencing, 16S rRNA is variously used for phylogenetic trees as well. So as you can see over here, 16S rRNA gene is conserved in bacteria, and it contains various hypervariable regions that can provide various species-specific signature sequences, and that is why 16S rRNA sequencing is a very popular sequencing technique. It is fast speed, cost efficient, and also has high precision. And this is also used in case of phylogenetic studies because it is conserved so properly in bacteria. So that is why 16S rRNA can be matched with phylogenetic studies. Phylogenetic tree. And then nitrogenase. I talked about nitrogenase previously only. Nitrogenase is inhibited in the presence of oxygen, and that's why free living diazotropes or free living nitrogen fixing bacteria need to protect the nitrogenase from surrounding oxygen. So nitrogenase gets inhibited in aerobic conditions in the presence of oxygen. So she's correct. It will be 1A, 2B, and 3C. Yes, the nitrogenase enzyme complex, the nitrogen fixing enzyme, it's sensitive to oxygen that irreversibly inactivates the enzyme. And diazotropes, that's why, must employ mechanisms which on the other hand permit the supply of oxygen required for energy regeneration, and this will protect nitrogenase from the deleterious effect of O2. Effect of O2. So this question, which of this is true? This is also from the lecture videos that was uploaded. These are very factual. If anybody wants to try, they can. These are very factual questions, so you need to have factual knowledge for this. Which of these statements are true? Anybody would like to try? This is from the lecture videos, so you can go back to the lecture video slides and also try to figure this out. I think it's from lecture 17, lecture 17 or lecture 18 slide. I'm not sure. I have to check. Anybody? Okay. So with these statements, thioplaca uses a nitrate as electron acceptor and sulfur as the energy source. This is a true statement. And thiomargarita are sulfur oxidizers and have large sulfur storing vacuoles. So yes, so this is thiomargarita structure. So it's a giant sulfide oxidizing bacteria, and cells are about 100 micrometer in diameter, and they have very large vacuoles. So you can see from the diagram here as well, they have very large vacuoles. So this option, thiomargarita sulfide oxidizers and large sulfur storing vacuoles, is correct. The slide is also from the lecture videos. And thioplaca is the type of bacteria that uses nitrate as electron acceptor and sulfur as energy source. This is also shown in the lecture slide videos, so you can go back and check this out as well. Next is, which of these are associated with torque? Anybody? So ATP synthesis, I think PMF should also be in the answer. PMF, proton motive force, is also let me show you. ATP synthesis is obviously associated with torque, and PMF, that is the proton motive force, it's also associated with torque. Wait, yes. So PMF is the proton motive force that actually allows the protons to be pumped inside the matrix from the intermembrane space via the ATP synthesis. ATP synthesis leading to the generation of ATP. So PMF also leads to torque, and obviously ATP synthesis, because the entire process, the main agenda of doing the entire process going through ATP synthesis. So that is one thing. So both proton motive force and torque should be both proton motive force and torque should be in this list. So next question is, I have shown the answer already anyway, but still, if anybody wants to try. Consider the following statements. Chemolithotropes grow faster than heterotrophs, so consumption of glucose gives the most favorable value of Gibbs free energy. Anybody? So only 2 is true. There's no scientific evidence that show that chemolithotrophs grow faster than heterotrophs. There might be one bacteria that might be growing faster than the other bacteria, but we cannot stereotype or generalize the entire category under that. So chemolithotrophs don't really grow faster than heterotrophs, but consumption of glucose gives the most favorable value of chemistry energy, definitely. The next question is, which of the following statement or statements are true for purple bacteria? Anyone?
Option B and C.
B and C. Okay. B and C, anoxygenic photogenesis, okay, exhibit Z scheme of energy excitation. Okay. So let's see about the purple bacteria. What is the purple bacteria first? So this purple bacteria, they definitely engage in anoxygenic phototrophy. That is, they do not generate oxygen during the process, and they have a single photosystem with bacterial chlorophyll, allowing them to use this cyclic phosphorylation as step here for the formation of ATP. But if a purple bacterium wants to grow as a photoautotroph, it will also need reducing power in the form of NADPH. Okay, and the reaction center of purple bacteria, it's shown here, it's called P870, and after being hit by a photon of light, the potential changes and it is insufficient to reduce NADPH. Thus autotropic purple bacteria must engage in a process known as reverse electron flow. So they also engage in a process called reverse electron flow, because the energy that is basically the potential changes when photon light hits the center, but still that is not enough to reduce the nicotinamide adenine diphosphate is NADP basically. So that's why they also engage in reverse electron flow when they are growing as phototroph, and by this they use energy from the proton motive force, the PMF, as I explained, proton motive force, to drive electrons up the electron tower. And additionally, they also need to find an external electron donor to replace the electrons now diverted to NADP. And typically these electrons come from hydrogen disulfide or elemental sulfur, with various sulfur byproducts produced, as we can see here. And in the presence of organic compounds, this purple bacteria also exist as photoheterotrophs, and in that case they utilize photophosphorylation to generate ATP, and they'll get their organic compounds from the environment. And basically this eliminates the need for using reverse electron flow, an energetically unfavorable process as well for the external electron donors. So the option 1 and 2 will be correct, that they can counter the reverse electron flow in photosynthesis, because when they are in the presence of organic compounds, they don't really need the reverse electron flow, because now they have organic compounds, now they'll have enough energy to be able to reduce NADP. Previously they were not having enough, they were only relying on the photon, the light, and that was not enough. But now if they are in the presence of organic compounds, I think that will be enough for them to reduce NADP, and they can then counter the reverse electron flow, and they can basically eliminate the need for reverse electron flow, which is an unfavorable process. Energetically it's not favorable. And the other one was anoxygenic phototrophy, definitely, because they do not generate oxygen during the process. So that the previous option was correct, they engage in anoxygenic phototrophy. So that's it. So these are the purple sulfur bacteria. So they carry out anoxygenic photosynthesis is also taken from the lecture slides. There's no oxygen involved. There are very morphologically diverse group. Their genera fall within alpha, beta, gamma proteobacteria. They can contain bacteriochlorophylls and even carotenoid pigments, and they produce intracytoplasmic photosynthetic membranes with varying morphologies. So we come to the end of the session. Anybody has any question from any of the problems that we discussed today? We can talk about that again, or we can end the session also. So this is how. So all these questions, the answers are mostly from the slide videos, the lecture slide videos only. So if you go to the lecture slides uploaded already on the NPTEL website, you'll be able to find the answer to most of these questions. And some of these questions are really not fact based, like factual questions, and factual questions are not that interesting to me, at least, but some questions are really conceptual, like gram staining and all that, which requires you to understand why the different types of bacteria take different states, why one bacteria why the stain gets washed out of one bacteria on adding alcohol, and why it doesn't happen to the in the other case, because of the difference in the composition of the cell membrane. So these things are quite fascinating, but there are also very factual questions, like in case of thioplaca, thiomargarita, that question. All of these are very factual. For that you have to go through the slide lecture videos also. But if you have any doubts in one of any of the concepts, conceptual questions, then please feel free to ask. If I'm not able to answer right now, I'll go back and check and let you know in the next class. Also, all of these slides will be uploaded, and all of this recording we have recorded this session, and I'll upload this session on YouTube as well. So anything that you would like to add? Anybody? Okay, I see some people were answering in the chat box also. When I'm presenting my screen, only the PPT is visible to me, so I'm sorry I am not able to see the chat box at all. So I might not have acknowledged your answers, but thank you for the session. Anybody would like to say anything, or we can stop the session? We'll meet again next Friday, same time, same link.
Thank you.
Thank you, sir. Thank you for saying talking about the electron microscope thing, because I really had no idea, but it was great to know about the electron microscope thing. Thank you so much.
Yes.
Anybody else?
My work is related to virology.
Oh, okay. Nice. That's great. Thank you so much. Anybody else? Then we'll meet next week, same time, same link. Thank you. I'll close the session today. Thank you.